Okay, this is the Chapter 14 lecture on host defenses. So in the next couple chapters, 14 and 15, we will learn about the host's response to a microbial infection. So there are systems in place which try to prevent microbes from becoming established, growing to high numbers, and making the host sick. And these systems are collectively called immunity. They are the host's defenses. And we have two arms of immunity. In Chapter 14, we'll discuss innate or nonspecific immunity, which is comprised of the first line of defense and the second line of defense. In Chapter 15, we'll discuss acquired or specific immunity, which is comprised of the third line of defense. The difference between innate and acquired immunity is that innate immunity is present at birth. It does not change throughout our lives. It's a very quick and immediate response to microbial invasion, but it is also general. So all microbes will be responded to in the same fashion by the innate immune system. Compare that to the acquired immune system, which is a much slower response and takes several days or weeks to build, but is very specific. So there's a tailored response to each individual microbe. So we'll start by talking about the first line of defense. Defined here are the two types of immunity, and also listed under them are phrases that mean the same thing. So synonymous with innate immunity, you may also see it referred to as natural immunity or nonspecific immunity. Adaptive immunity you can also call acquired immunity or specific immunity. So let's focus in on innate immunity, the first line of defense. So initially, the first line of defense's job is to prevent microbe entry into the host. So one of the things that help to do this are the normal microbiota, which is the term collectively referring to microbes that grow normally in a healthy person. So in this individual shown, we see the normal microbiota colored in green, and they are preventing growth of disease-causing or pathogenic microbes by competing for space and resources. And this is called microbial antagonism. So the normal microbiota prevent the disease-causing microbes from becoming established through microbial antagonism. We also, in the first line of defense, have several physical, anatomical, and chemical defenses that prevent entry of microbes into the body. So some examples of physical or anatomical barriers are the skin, which is a collection of epithelial cells that are very tightly bound together and create a very impenetrable barrier that will be difficult for microbes to get past and get into the host. Lining cavities like the respiratory tract and the gastrointestinal tract that are open to the outside. We have mucous membranes, which are also a connection of very tight cells. They secrete mucus, which is an antimicrobial substance that can trap microbes that are inhaled or that get into the gastrointestinal tract. And these microbes, once they're trapped in mucus, will be eliminated from the body, either by coughing them out or by excreting them in the urogenital system. In the first line of defense, we also have several non-specific chemical defenses. So some examples of these are tears in the eyes, which contain an enzyme called lysozyme. It's also present in saliva and sweat. And lysozyme will break down the cell wall of bacteria. We have sebaceous glands, which release oils in the skin that are antimicrobial. Earwax is antimicrobial. Sweat, which causes the pH of the skin to drop and for the skin to be salty, will prevent growth of microbes. And then in the stomach, we have a very acidic pH where most microbes are unable to grow. So we generally have a release of substances like urine, feces, sweat, earwax, and tears, which will also prevent entry of microbes into the body. Listed here are other mechanisms in the first line of defense. Their collective goal is to keep microbes out of the host. If a microbe is able to penetrate the first line of defense and enter the host, so it gets past these physical and chemical barriers, we then see activation of the second line of defense, which is still part of innate immunity. But the second line of defense will be a response to the microbes inside the host. So second line of defense main players are phagocytes, we have inflammation and fever, and then we have the antimicrobial proteins called complement. Important for the second line of defense is the lymphatic and circulatory systems because these connect all cells and tissues in the body. So the circulatory system is our system of vessels which circulates oxygenated blood throughout our bodies. The lymphatic system, shown in green, closely mirrors the circulatory system and serves as a filter for the liquid portion of the blood. So in the lymphatic system, we have a lot of immune detectors like phagocytes, which will be present and will attempt to locate any invading pathogens and communicate their presence to other parts of the body and other immune cells in the body. The lymphatic system is made up of primary lymphoid tissue, which is the thymus and the bone marrow, and these are areas where cells of the immune system, namely the B cells and the T cells, will mature, and we'll learn more about B and T cells in chapter 15. So these cells mature in the primary lymphoid tissue, and in the secondary lymphoid tissue, they will encounter foreign invaders like pathogenic microbes. And all of these foreign invaders, pathogenic microbes, other things that activate the immune system are called antigens. So the B and T cells mature in the primary lymphoid tissue and they encounter antigens in the secondary lymphoid tissue, and then they can orchestrate a response by the third line of defense in the immune system. One of the main players of the second line of defense, so these will be an early responder cell if a microbe gets into the host, are the phagocytes. So the phagocytes are charged with surveying the tissues, destroying microbes, and recognizing antigens, which is this term to mean any foreign molecule that initiates an immune response. So in this very complicated figure from your book, it's showing you that all of these cell types that are important in the immune system are derived from stem cells. I've put boxes around the ones we're focused on in the second line of defense: the neutrophils, the basophils, the macrophages, and the dendritic cells, because these are all cells that are called phagocytes. So they have the ability to locate and destroy foreign invading microbes. So in this figure, I would just like you to be familiar with the types of cells that are phagocytes, but you don't need to know where they derive from. Shown in this figure is a phagocyte in the lower right corner. We have a microbe, which is drawn as a very short blue bacillus, so it's kind of an oblong shape, and there's a little cluster of them in the upper left corner of the figure. So we have the phagocyte next to the bacteria, and then an up close image of that in the lower right corner on this figure. So in phagocytosis, which is how a phagocyte responds to a microbe, the first step is chemotaxis, and so this is the phagocyte following a chemical trail to find the invading microbe. So this is how the phagocyte finds the microbe. The second step is adhesion of the phagocyte to the microbe. So in this step, we have on the surface of the phagocyte receptors called pattern recognition receptor. So these will be able to bind to patterns on microbes or foreign molecules. On the surface of the bacteria are pathogen-associated molecular patterns. So the pathogen recognition receptor on the phagocyte binds to the pathogen-associated molecular pattern on the surface of the microbe. So these two are shown bound up in step 2. Once the microbe is bound to the surface of the phagocyte through this interaction, in step 3, the microbe is engulfed into a vacuole, so it's now in the interior of the phagocyte. And in this structure, in step 4, the microbe is contained within this vacuole, and that structure is called a phagosome. Step 5, inside the phagocyte, the phagosome, which contains the microbe, will fuse with the lysosome, which contains enzymes and reactive oxygen products. And all of these enzymes and reactive oxygen products will destroy the microbe so that it is degraded, as shown in step 6. And then in step 7, debris from this degradation of the microbe is released outside the phagocyte. So the phagocyte, this cell of the second line of defense, has successfully found the invading pathogenic microbe, engulfed it, and destroyed it. So it's now cleared it from the host. The interaction between the phagocyte and the foreign invader is shown in this figure. So this specific type of phagocyte is a macrophage, and it's coated in pattern recognition receptors, and one type of these is the toll-like receptor. So the surface of the phagocyte has the toll-like receptor, which is going to bind to the pathogen via interaction with pathogen-associated molecular patterns, or PAMPs. An example of a PAMP would be peptidoglycan in the bacterial cell wall. There are several ways that bacteria have evolved to prevent phagocytosis. So one of them is to simply kill the phagocyte by releasing phagocytic destroying molecules. So two microbes that do this are Streptococcus pyogenes and Staphylococcus aureus. Microbes have also evolved ways to prevent chemotaxis, so the phagocyte cannot locate the microbe. Bacteria with capsules are able to block the adherence step, so the pattern recognition receptors on the surface of the phagocyte cannot bind to a capsule, so they cannot adhere to the microbe. Some microbes will get pulled into the interior of the phagocyte, but then they can escape from the phagosome and remain inside the phagocyte, residing in the cytoplasm. Some microbes can prevent formation of the phagolysosome, which is what's formed after the phagosome fuses with the lysosome. Another part of the second line of defense is inflammation. So this is shown in figure 1418 from your book. We have in step 1 an injury, which introduces bacteria into the host. We have in step 2 the vascular reactions, which we can discuss in more detail with the words listed in red. So in the vascular reactions, we have an initial constriction of the vessels until the wound is closed off to prevent loss of blood. We then have a vasodilation, which is when the vessels become dilated and larger, and that's in order to bring blood containing immune responders like phagocytes to the area. In margination, the immune cells brought by the blood are able to stick to the walls of the vessel around the area of the injury. In diapedesis and extravasation, the immune cells are able to squeeze out of the vessel into the surrounding tissues so they can deal with the bacteria that's been introduced into the host. Chemotaxis is the release of chemicals to recruit more immune cells to the injury, and phagocytosis would be these immune cells engulfing and destroying the invading microbe. In step 3, we have edema and pus formation, which is a result of death of phagocytes as well as death of the microbe. And then in step 4, we have resolution, so most of the microbe has been cleared from the wound. We have a scar formed and the vessel has been repaired. Often accompanying inflammation is fever. So fever is caused by the hypothalamus getting reset to a higher temperature. So the hypothalamus controls body temperature of the host, which is normally 37 degrees centigrade. But during fever, molecules called pyrogens are released by white blood cells. They circulate in the host and they cause the hypothalamus to increase the normal set body temperature of the host so that the host body is at a slightly elevated temp. The benefits of fever are threefold. We have the outcome of induction of production of host iron-binding proteins. Two examples of iron-binding proteins that humans can make are transferrin and lactoferrin. Both of these molecules reduce circulating free iron in the host, and iron is an essential nutrient for microbes. So without free iron, microbes cannot reproduce and replicate. Fever is also thought to slow growth of some microbes, and it seems to increase the performance of the immune system and ability of the immune system to respond. The last arm in the second line of defense is the complement system. So this is a system of antimicrobial proteins that are activated, and their outcome is destruction of a microbe. The first and second lines of defense in Chapter 14 are part of the innate immune system. In Chapter 15, we will discuss adaptive immunity. The complement system is part of innate immunity, but is activated by adaptive immunity. So in this way, it kind of bridges innate and adaptive immune systems together. Shown in this figure is the three ways that the complement pathways can be initiated. Complement pathways are orchestrated by several proteins that are produced by the liver and circulate throughout the host in the blood. And these proteins are named C1 through C9. Each of the complement pathways shown here is a cascade, where one event occurs and that leads to the next event in the cascade, which results in the third event in the cascade, and so on. So there are three ways that complement pathways can be initiated. I'll describe the first one in detail, although for your exam you just need to know how they are initiated and what the outcome is. So in the first complement pathway, called the classical pathway, it is initiated by an antibody binding to an antigen. We will learn about antibodies. They are part of adaptive immunity, which we'll discuss in Chapter 15. Antigens are foreign molecules that activate an immune response. So the adaptive immune system makes an antibody against an antigen, which binds. This causes complement protein C1 to interact with the antibody. As a result of that, complement protein C2 and C4 are split into two components. C2a and C4b will then bind together, which cause complement protein C3 to split into two components. So the classical pathway is initiated by antibody binding to antigen. The alternative pathway is initiated by the presence of lipid carbohydrate complexes in the host that are only present on microbes, so they are not present on host proteins. So the presence of these lipid carbohydrate complexes signifies that a microbe is invading the host, and this will initiate the alternative complement pathway. The third complement pathway is initiated by carbohydrate molecules containing mannose, which also are only present on microbes. As a response to the presence of mannose, there is a molecule made by the host liver called lectin, and this will initiate the lectin pathway. So each of these three complement pathways are initiated in different ways. The outcome of all three is the same. So the outcome of the classical pathway could be inflammation, cytolysis, or opsonization. The outcome of the alternative pathway could be inflammation, cytolysis, or opsonization. And same with the lectin pathway. The outcome, once this is initiated, is inflammation, cytolysis, or opsonization. So on the next slide, I'm going to show you what these three outcomes mean. So any of the three complement pathways can result in these three outcomes. The first outcome is called cytolysis. Together to form a membrane attack complex, which is essentially a hole punch that will cause holes to be created in the surface of the